Welcome to the Center for Evolution and Medicine seminar series. Today, it's my pleasure to introduce a close collaborator and our speaker, Daniel Promislow from the University of Washington. So Daniel began his career in biology as an undergraduate at UChicago, got his PhD in evolutionary biology at Oxford, and after some postdocs was a professor first in genetics at the University of Georgia, and for the past 9 years, now professor of laboratory medicine and pathology and biology at the University of Washington. Daniel's broadly interested in evolutionary genetics with a particular focus on projects related to aging, as well as studies on sexual selection, natural genetic variation networks, and metabolomics. Daniel wrote a bunch of wonderful comparative evolutionary papers on theories in the evolution of aging across species, like Living Fast and Dying Young, which is a comparative analysis of life history variation among mammals back in the 90s, and then some more work on that in birds later on in that decade. Daniel leads the Dog Aging Project, which is a really large NIH-funded project, citizen science project, community science project on companion dogs, which I'm sure many of you are familiar with and some of you even work on. Daniel is Daniel is also an unbelievably wonderful mentor, not only to undergraduates, graduate students, postdocs, but also I can personally attest to as to junior faculty members like myself and many others. So he's very excited to meet with many of you while he's here, but I'm sure he's also happy to hear from you afterwards if he doesn't get to meet with you some more. I ask Daniel for some fun facts about him because I think it's nice to make this a little bit more personal and less about academia always. And the one personal thing I knew lots of personal things about Daniel, but one thing that he told me, right, so one thing that I can share with this audience here is that Daniel actually recorded an album at some point a couple decades ago. Yeah, right. I don't know if he had any groupies. I don't think he did. Daniel was the rhythm guitarist in a Celtic band called Short Road Home, who had a self-titled album, Short Road Home. And with that, I think I'm just going to leave it to Daniel to take it over, and no music today. But everyone, please welcome Daniel Promislow.
Thank you, Noah, for that very kind introduction, and for the invitation to speak here. I'm really pleased to be here. I've known about the center for a very long time, really since its inception. I remember when it started, and so it's really a pleasure to be here. And I look forward to a lot of conversations while I'm here. So I'm going to talk about a couple of things today. First, I'll spend a little bit of time just briefly sharing why I think aging is a worthwhile phenotype to work on. And then I'm going to tell you about work that I've been doing the people in my lab, I should say, have been doing over the past decade or so, using metabolomics as a way to really understand the genotype-phenotype map in aging and age-related traits. And then I'll spend a little bit of time just telling you briefly about the Dog Aging Project. We are always recruiting collaborators as well as participants if you or a friend or a family member has a dog. So I have to tell you a little bit about the Dog Aging Project. So let me just start first talking a little bit about aging and why aging is a worthwhile thing to measure. Well, first of all, from a health medicine perspective, most of the causes of mortality in modern human populations are tied in with aging. So if we look at the major killers in this country, heart disease, cancer, neurodegenerative disease, we know a lot about risk factors from many, many decades of epidemiological studies. Diet, lack of exercise, smoking, infectious disease, all of these things, even very early in life, can impact risks later on. Now, if we put a couple of decades of aging on the same plot, so all the risk factors are together, what you can see is that a couple of decades of aging will dwarf the effects of all these other risk factors. So from that perspective, if we can understand what it is that leads to age, increasing the risk exponentially for heart disease, cancer, Alzheimer's disease. The risk doubles about every 8 years for heart disease and cancer, doubles about every 4 years, though it starts later, for Alzheimer's disease and Parkinson's disease. So if we could understand why that is, we could really have a dramatic, potentially dramatic ability to either delay or ameliorate the effects of aging on these major killers. What actually draws me even more to the study of aging is that it's a kind of conceptual hub for a way of knowing the world, and on diverse scales. So aging connects spatial scales. If you look on the left, we can think about studying molecules and how concentration of molecules, like the metabolites that we work on, change with age, all the way up to how age impacts mortality in whales and redwood trees. So we can study aging on a spatial scale over more than 10 orders of magnitude, but also on a temporal scale. We can think about aging of biological phenomena that are happening on a very rapid temporal scale. So we can study something like VO2 max. If you look at a trait like nerve conductance velocity, so very rapid trait, interestingly, it shows almost no effect of age. Don't know why. So there's some things that age quickly, some things that don't. At a demographic level, aging can be measured in some organisms in terms of hours. So this is a mayfly. Mayflies live for about a day. There's some nice work that Jim Carey did in the field where he captured mayflies as they were emerging and then tracked them hour by hour, and mortality rates increase with age if you age them hour by hour. So a 20-hour-old mayfly has a higher mortality rate than a 2-hour-old mayfly. And then at the other extreme, this is one of the longest lived animals, this ocean quahog clam. Individual clams have been shown to live more than 500 years. Of course, to do that, they had to kill the clam and cut the shell. And then, of course, at a vast temporal scale, we can compare patterns of aging across these species and try and understand why it is that some species are so much longer lived than others. So aging really captures these spatial and temporal scales, and I'm not going to talk about it today, but we can also use aging as a conceptual focus well beyond biology. Any of you could have a conversation with an artist like Rembrandt painting self-portraits as he aged, or a sociologist or an economist. It really is this focus for a broad conceptual lens on the world. In the past 30 years or so, geroscientists, people working on the biology of aging, have figured out how to make lab organisms live longer, either through genetic manipulations or drug or diet interventions. Quite dramatic. And almost all of this work has been done in yeast, worms, flies, and mice. And of course, when we're training students in our labs, we teach them that controls are essential. And in this case, usually that means using organisms that are genetically inbred, so no genetic variation, and then controlling the incubator environment so that there's no environmental variation, and then manipulating the one thing that we're interested in testing. And when we do that, we can make these organisms live dramatically longer. And for about 30 years, I've been going to aging meetings where my colleagues have said, and someday, maybe this will get into clinical trials. And I'm not going to talk about this today, but we're actually at a really fascinating time because we are now in an era of clinical trials. There are many clinical trials testing some of these drugs in people. We are doing this in dogs, and I'll tell you a little bit about that right at the end. So here's the challenge. The real world is very different from the lab. People are not inbred, and people don't live in a single incubator. There's lots of variation. And there's this classic picture that's shown in quantitative genetics textbooks. These are cadets at Connecticut Agricultural College back in 1914, lined up from shortest to tallest. They form a nice bell-shaped distribution. if the undergraduates at Arizona State, and I understand there are more undergrads here than just about any school on the planet, if they were to line up, they would make a nice bell-shaped curve as well. And all of that variation would be due to two and only two factors, genes and environment, and the interaction between the two. And that's that equation there. So phenotypic variance is the sum of genetic variance, environmental variance, and the interaction between the two. And this is our tremendous challenge. How do we get out of the lab and into the real world and understand what those elements are? How do we decompose the genetics and the environment? That's the great challenge, and many of you in this room are working on that question. So rather than that equation, we can show this as a picture, and this will come up a couple of times. And so this is really what I've spent a good chunk of my career doing initially, as Noah mentioned, thinking about phylogenetic variation, but really since about 1995, 94, when I started working on Drosophila, thinking about quantitative genetic variation. And then more recently, over the past 10 or 12 years, trying to understand not only what the genes and environmental factors are that shape aging, but what the underlying mechanisms are by which genes and environment shape aging. And there are lots of different mechanisms that one could explore. In my own work, I focused on the metabolome. It's a little bit hard to see in this picture. The picture on the left shows the structural elements that make up the metabolome, amino acids and lipids and nucleic acids and so on. And then in the middle, there's the Krebs cycle. So the cool thing from my perspective of the metabolome is that these are the structural and functional building blocks of all life. Every organism, from archaebacteria to that redwood tree, are made up of metabolites. And so really what we're doing in my lab is trying to understand these interactions between genetic variation, sometimes environmental variation, metabolomic variation, and aging. And I'll come back to this, but one of the motivations is that the metabolome is highly conserved. So a glucose molecule in a Drosophila is the same glucose molecule in us. And so if we learn about glucose metabolism in flies, we're likely also going to be learning about glucose metabolism in us. So I'm just going to tell you a few different stories about how we've used the metabolome to kind of parse this equation. And the first argument that I want to present is that the metabolome actually is a really powerful bridge between genotype and phenotype. So here, for example, I'm showing you data from a study that came out a couple of years ago from Kelly Jin, who was a graduate student, looking at the effect of diet restriction in a set of about 200 inbred lines. So some years ago, Trudy McKay, who was at North Carolina State at the time, went to the Raleigh Farmers Market, collected a bunch of female flies, brought them into the lab, and did full SIB inbreeding for 20 generations. And so all of these lines, hundreds of lines, are effectively like inbred mice or inbred dog breeds. And she then did whole genome sequencing, and people have obtained this panel of flies. They're in labs around the world. People are measuring different traits and then mapping them using genome-wide association studies like one might do in humans. So what I'm showing you here is the range from genotypes that when put on diet restriction live about 20 days shorter, all the way to the right side where diet-restricted flies in that genotype live about almost 30 days longer. So diet restriction is the one environmental manipulation that is most consistently shown to extend lifespan in mice, yeast, worms, and flies, and even in macaques and non-human primates. But with the exception of just a couple of other studies, these diet restriction studies are always done in a single genotype. So we decided to do this in 170 or so genotypes, 160 something, and then to try and ask, well, what's going on here? So we looked at genetic variation, and we didn't actually find any genes associated with this response from shorter lived to longer lived. we then did metabolomics on all these flies, on ad-lib food and diet-restricted food. And what you're looking at with the orange circles, each one of those is a metabolite. And the lines connecting the orange circles to that gray circle in the middle are drawn when a metabolite is significantly correlated across all these lines with how the line responds. So, for example, levels of alpha-ketoglutarate are, in all these lines, are associated with whether the line is longer-lived or shorter-lived. And the point I want to make here is those green circles are specific genes. So this one over here on the left, CCHA2R, actually is it's not significantly associated with the diet response, but it is significantly associated with each of these 5 metabolites, which in turn are associated with the diet response. And in fact, we then went and knocked down CCHA2R and found that it did indeed alter the diet restriction response. So this was work we did in collaboration with Pankaj Kapahi at the Buck Institute. So the metabolome here is literally acting as a bridge between the phenotype and the genotype. The genotype effects are quite weak. And so, like in genome-wide association studies in humans, it's hard to identify them. The metabolome effects are quite large relative to the genes, and so really it kind of magnifies that genetic effect and allows us to bridge those together. More recently, this is work not yet published. We've been looking at using the same approach to look at sensitivity to rapamycin. Rapamycin is the drug that is most successful at extending lifespan in mice. There are also data showing that we can do it in yeast and worms and flies. And there are now clinical trials in humans and trials that have been published showing an improvement in immune function in older humans treated with low-dose rapamycin. So I'm showing you plots from 2 different strains. So here's a strain where, and this is not aging, but time from egg to pupa, so development time. And as expected, rapamycin slows development time quite dramatically. But there are strains that are completely resistant to the effects of rapamycin. And here they all are, lined up from a set of strains on the left that show no response to rapamycin to ones whose development time is almost doubled from about 8 days to 15 days or so. And so we then picked out some sensitive lines and some resistant lines at the extremes and did metabolomics. And when we did that, we found 2, and again, very weak, if any, genetic differences between these resistant and sensitive lines. When we do the metabolomics, so what you're looking at here in the pink and sort of light and dark pink are sensitive strains, and in blue, light and dark blue are the resistant strains. And these are plotted on an axis of principal components. So each dot represents a sample of 5 flies extracted from metabolomics. So we take the metabolomic data, which are about, in this case, I think about 125 or 130 different metabolites, and we collapse them into 2 dimensions using principal components. And you can see that almost fully separates the resistant and sensitive lines. But there's something else that we see as well that's kind of interesting, which is that the sensitive lines treated with RAPA look different metabolomically than the sensitive lines that are on control food, whereas for the resistant lines, there's no effect. So what's going on here? Well, we don't know. But we can speculate that one possibility is that the metabolome is actually upstream of growth, and for some reason, when we treat flies with rapamycin, which downregulates a pathway called the mechanistic target of rapamycin pathway, for some reason, some lines have a robust metabolome that doesn't change, and so there's no change in larval growth. Alternatively, these 2 are independent and something else is going on. For example, there might be something upstream that resistant lines have mTOR that's resistant to the effect of rapamycin. And of course, we're going to go back and be looking at both transcriptomics and proteomics to see what's going on with mTOR. But here's the really interesting thing. If we look at other studies we've done, so this is published work from Ben Harrison, who's a senior scientist in the lab, where we treated flies with peroxide and looked at how long they lived between 2 and 5 days. And again, we've got sensitive over here and resistant over here. The sensitive are significantly different. The resistant are not. And then here's a completely separate study done in collaboration with Scott Pletcher's group, where flies are put on 2 different diets. One is a mixture of protein and carbohydrate. The other has separate wells for protein and carbohydrate, and they have to choose how much they want. When they choose, they don't live as long. It's actually a stressful environment for them. And if we look at the metabolome, the ones on the choice diet have a different metabolome than the ones on the standard diet. And that's altered depending on whether you knock down serotonin or not. So there's something not only about metabolomic variation tying phenotypic response, but also the changes in the metabolome seem to be associated with whether or not there are changes in the phenotype in a genotype-dependent way. So there's really something interesting going on. And we haven't yet done the mechanistic work to try and figure this out, but I think there's something really interesting here. So within the aging field, starting about 10 years ago, people started getting very excited about the ability to predict how old a person is based on their epigenome. There were 2 papers that came out almost 10 years ago. One from Trey Eideker's group and the other from Steve Horvath. Both of them showed that if you look at genome-wide patterns of methylation and choose a subset of those sites, a few hundred, you can very accurately predict how old someone is. And that work, those 2 papers, spawned a cottage industry, driven, I think, in part by the excitement of the work and in part by how excited those folks were to convince everyone else to start building epigenetic clocks, methylome clocks. And so there are now hundreds of papers in the field. The epigenome, if you think about the sort of conceptual biological distance from genotype at the top to phenotype at the bottom, the epigenome is way up here, but I think the real action is in the metabolome, the lipidome, the building blocks downstream closer to phenotype. So we thought, well, let's build a metabolome clock to see if we can predict age with the metabolome. Now, we're not the first to do this, and it's actually been done in humans. So this is a plot showing chronological age in humans versus showing predicted age versus chronological age. And here, the predicted age is derived by putting all these metabolites into a statistical model and trying to predict age of the individuals from which the sample was taken. And in this case, you can see there's a pretty good, highly significant relationship with an R-squared of over 0.4 between the predicted age and the chronological age. And what the original methylome papers suggested, and is suggested here as well, is that if you look older than you actually are, well, maybe you're sicker or you're not going to live as long. And in fact, that turns out to be the case. So if you look at probability of survival and you compare the first quintile and the last quintile, it turns out that they're significantly different, that individuals that are well above the line tend to be shorter lived than individuals that are below the line. And here's another study doing the same thing, showing that you can actually predict 5-year survival from the residual variation of this model. So the problem with humans is that it's hard to do experiments on people. You need really big incubators, and they just won't fit. So we thought, well, let's turn to the fruit fly. So what I'm showing you here are samples from 20 different genotypes colored by age. So we sampled the same 20 genotypes at each of 6 different ages. And this is the first principal component axis of the metabolome. So each one of these samples is the metabolome of a different genotype. And what you can see is that we can clearly differentiate the young samples from the middle-aged samples from the old samples. Here, flies living up to day 80. And in fact, we can predict the age of a sample. And what we find is that ones that are above the line, that look older than they should be, have a shorter mean lifespan at day 45. So we actually can build a metabolomic clock. We've done that. And then we thought, well, maybe we can apply our clock to other experiments. So we started with our own experiment, which was done on the same flies, but a few years earlier, where we'd measured the metabolome. So it turns out that the predicted age of flies under diet restriction is younger than flies on ad lib food. So that makes good sense. We think that diet restriction makes mice and flies and worms younger, seem younger, and at the metabolome level, that turns out to be true. We then tried, this was a harder, sort of a higher bar. We went to another lab that had done some experiments on a completely independent set of flies. So this is work from Mark Phillips, who was working with flies derived from Michael Rose's experiments that go back to the early 1980s, where he selected for long lifespan in flies. And so the long lifespan was selected by delaying reproduction. Here are the delayed reproduction flies. Here are the flies that turn over rapidly. And completely different flies, different lab, fewer metabolites that overlap because they used a different system. And still we were able to show that the long-lived flies look younger than the short-lived flies with this metabolome clock. So this is work not yet published. So we're excited to keep on developing this metabolome clock. So and then the final thing I want to say about metabolomics is that I actually think there's tremendous opportunity to take this metabolome perspective to really think about pretty deep evolutionary questions. So as you know, there are now decades of work on genome evolution. People have thought long and hard about how genomes evolved. How is it that there are whole classes of genes that are lost over evolutionary time and gained over evolutionary time? Many people have done this in fruit flies, of course in other species as well. But what about these building blocks? We know that there are metabolites that don't change over time. Glucose, for example. There are a lot of metabolic pathways that are in E. coli and are in us. So there are lots of features that don't change, but the levels of these metabolites and how they correlate with each other, how they form modules, could evolve over time. It's also likely that there are thousands of metabolites that are unique to different organisms that we don't know about because we can't find them. Unlike genetics, where we can sequence the genome, if we don't know a molecule is there, we may find a peak on a mass spec, but we'll have no idea what it is. And defining one molecule can take many months of a chemist's work. So it's really kind of the Wild West in metabolomics. We don't know a lot about what's out there yet. But we thought, well, let's start with a well-studied group of organisms. So these are fruit flies. These were mostly among the first flies that were sequenced when people started sequencing whole genomes in flies. And we brought them all into the lab. We reared them in a common environment. So it's a common garden study. They were all on the same food. And we measured their survival. So here I'm just showing you their survival curves, proportion alive on the y-axis as a function of days. And you can see there's one species that lived about 170 days. The students working on this project were not happy about that one because they wanted to go home for Christmas. And we measured metabolomics from all these strains as well at 5 days and at 31 days. We did both targeted and global metabolomics don't have time to go into the details, but I'd be happy to talk with you about that afterwards. And so just a couple of interesting findings. So just like we can do with the genome, we can, with the metabolome, we can ask, well, how do these metabolome patterns diverge over time? And how does that relate to evolutionary divergence? And what we found was that the best model, we compared a few different models, and the best model appeared to be one where the divergence is linear over time, suggesting that at the level of individual metabolites and how much of each metabolite we see, they're kind of drifting over time. They don't appear to be under strong selective constraint. When we look at the level of modules, and again, I don't have time to go into all the details, but the paper came out last year. If you look at modules, so how the metabolites are interacting with each other, they do seem to be under some selective constraint. So this is really just the beginning. There's virtually no literature on this. And I really think there's a lot of opportunity here to think about how physiological function and failure to function evolves by thinking about the evolution of the metabolome. If we take these metabolites and we project them into this two-dimensional principal component space, there are some really clear patterns. So the first pattern that we almost always see in the metabolome, the strongest pattern is between males and females. So the females are on top in orange and the males down below in blue. But also we see a strong effect of age. The darker blue and darker yellow to the right are older, and the lighter to the left are younger. But as you can see, there's lots of variation as well, and that is phylogenetic variation. We can use phylogenetically independent contrasts to identify metabolite features that are significantly associated with lifespan. And we actually do this at the modular level, and so we identify different modules associated with longevity. After false discovery rate, none of them were significant. I think we're underpowered. But the most significant one, or the closest to significance, turned out to be a KEGG pathway called longevity regulating pathway. So it was not statistically significant, but it was reassuring that we're on the right track, and I would love to see someone do this on a larger scale. So I've already mentioned that the metabolome is highly conserved, and that was one thing that got me thinking that, well, maybe we should look at a more evolutionarily distant species from flies and maybe something closer to humans, and that led me to dogs. The project was inspired by a paper that came out in 2007 from Elaine Ostrander's group. It was a paper in Science on the genetics of size. You might have seen it. The cover of Science had a Chihuahua and a Great Dane walking side by side, so they differ by a factor of almost 50 in size. And about 40% of the variation in size in dogs is due to allelic variation at a single gene. So if you think about beautiful history of studies in humans on the genetics of size, and every time larger studies are done, we find more genes, and each gene contributes the tiniest amount. So there's one gene in dogs that accounts for a huge amount of the variance in size, is insulin growth factor 1, or IGF-1. So I saw that paper, I saw the headline, and immediately I thought, well, IGF-1 is associated with aging in mice. The pathway is associated with aging in flies and worms. Maybe the reason that large breed dogs are short-lived is because of IGF-1. So I read that paper and don't have time to tell you the whole story, but one thing led to another, and I got to have this wonderful opportunity to become a colleague with Noah and work on aging in dogs. So what I'm just going to tell you about briefly is what we're doing, and to invite you to reach out to me to join us. This is, Noah mentioned that this is a community science research program. We have 41,000 people around the country that have signed up their dogs, but it's also an open data, open science program. All our data are online, and all of you are welcome to work on it, and I hope you will. So the other inspiration for this study that I will show you when my spinning wheel stopped spinning. There it is. So this is Frisbee. Frisbee was a mixed breed 45-pound dog who died this summer. We had to put her down. Here she is a few years ago. That's Mount Rainier in the distance. We're at about 4,000 feet here. And when we used to go hiking with Frisbee, even when she was 10 or 11 years old, she would hike 3 times the distance because she would run ahead and run back and run ahead and run back. Really an extraordinary dog. At 14, she got pancreatitis, and she lived with it for 2 years. Here she is at 16 with cataracts, hearing loss, lipomas, as you can see, musculoskeletal difficulties. So why I would characterize her as an extremely healthy ager. For a 45-pound dog to live past 16 is really extraordinary. So how is it that dogs survive that long? Well, why study dogs? There are lots of reasons, besides the fact that we love dogs. They are the most morphologically variable species of animal on the planet. A scientist who works on our team likes to say that dogs are the broccoli of the animal world. So if you think about broccoli and Brussels sprouts, and they're all the same species too. So lots of genetic variation, lots of environmental variation. The figure on the top left, here's a dog in front of its doghouse, and then here are some dogs in their home. So there are rich dogs and poor dogs. There are healthy dogs that like to go hiking and not so healthy dogs that apparently like to smoke. There's tremendous variation in what dogs die of. The spectrum of diseases is much greater than any kind of silos that we can put people into. So what I'm showing you here are 4 different colored bars, blue, red, yellow, and purple, for 4 different species. So the first bar is respiratory disease, and you can see that bulldogs, the most common cause of mortality is respiratory disease, and that's generally true of the flat-faced or brachycephalic dogs. These little guys, Chihuahuas, most common cause of death is heart disease. Interestingly, dogs don't get cardiovascular disease, heart attack and stroke. So they get heart failure, cardiomyopathy, mitral valve disease. They don't get heart attacks like we do, nor do they get strokes. Retrievers get a lot of leukemia, lymphoma. And then finally, Shar-Peis have stomach and intestinal diseases. They're a really interesting breed. They have very high levels of hyaluronic acid. My colleague Vera Gorbanova at Rochester argues that the reason that naked mole rats, which are this big, a little rodent, can live for 30 years and not get cancer is because of their high levels of hyaluronic acid. We don't yet know if Shar-Peis are protected by their high levels of hyaluronic acid, but it's something we're interested in looking at. So just to wrap up, I'm going to tell you quickly about the structure of the project and what it is that we're doing. So the overarching goals are 2. First, to identify genetic and environmental determinants, just as we're trying to do in flies, of healthy aging. And then second, to see if we can actually intervene to increase healthy aging in dogs. So it's a long-term longitudinal study. It's funded by U19. This is actually an older slide. We are in year 5 of our 5-year grant. The renewal was submitted, and it will be reviewed in 4 days. 4 days. So cross your fingers for us. And here's that model again. Genes, so we have whole genome sequencing for about 10,000 dogs, as well as breed information that we get from the owners. Lots of environmental information, including EPA-provided air quality, carbon monoxide, sulfur dioxide, PM 2.5, PM10, those sorts of measures, green space, socioeconomic status, by zip code, by voting district, tons of environmental information. And then all the downstream data. And then, of course, in the middle, we've got that mechanism data, the omics. So here at Arizona State, NOAA is running samples to look at the epigenome and the microbiome in collaboration with Elchanan Borenstein at Tel Aviv. In my lab, we're doing the metabolome. We're looking at fluorescence-activated cell sorting, lots and lots of data. So there are 4 projects that make up the study. Project 1 is really creating geriatrics for dogs. Veterinary medicine has the same specialties that we do: orthopedics, ophthalmology, dermatology, you name it. No geriatrics. So when an old dog comes into a veterinarian, they've not had any training in what normative aging looks like in a dog. They have experience, but they don't have formal training to say, this is a healthy ager and this is not a healthy ager. So we're trying to create that. Project 2 is mapping that variation. Project 3 is understanding the mechanisms by which all this variation shapes healthy aging and developing clocks. And then Project 4 is an intervention study. We call it TRIAD. It stands for Test of Rapamycin in Aging Dogs. And it's a clinical trial. NIH doesn't call it formally a clinical trial because dogs aren't people, but we're running it like a clinical trial. So just a little bit more about the dogs who have joined us so far. So over 41,000 people have filled out the intake survey, which is about 2 hours worth of questions. And once they complete that survey, they become a member of the pack. I hope to get to 100,000. More dogs means more power to ask more questions. Within that group, we then have sampled cohorts. Foundation is 8,500 dogs. These are all dogs that are going to have cheek swabs for whole genome sequencing, and we're about 3/4 the way there. Precision makes up 1,000 dogs. We send the owner a kit to bring to the veterinarian to collect blood products, hair, fecal sample, and urine. And we're almost completely enrolled. And we've collected almost 800 samples from almost 800 of those dogs. And we're running those samples right at this very moment. And then the intervention cohort will eventually be 580 dogs. That's a harder process to enroll the dogs. They have to go mostly to veterinary teaching hospitals, and harder to get the right dogs and to get them in. And then there's some other studies. There's a brain health study and a centenarian study where we'll do some deeper genome sequencing. This is just to say we have dogs from every state in the country, including Alaska and Hawaii. And just to tell you a little bit very quickly about a couple of initial results. So the first thing that I wanted to know when these data started coming off the pike was, are they actually real data that are analyzable? And I thought, well, the first thing that ought to change with age is activity. I showed you that picture of Frisbee. And sure enough, owner-reported dog activity declines with age. Interestingly, the red line is rural and the blue line is suburban and urban. And what you can see is this is duration spent outside. And perhaps not surprisingly, rural dogs spend more time outside than urban and suburban dogs. The thing that did surprise us is that older owners say that their dogs spend a lot more time outside than younger owners. So this is owner reported. We will be putting actigraphy monitors on the dog to ground truth it. But initially we were surprised, and then we thought, well, maybe that makes sense because older individuals have more flexible schedules. The one exception was that you can't see here is people between 18 and 24 also were higher up, and that makes sense. Students have more flexible schedules. So we're just starting to map traits, and this was kind of cool. The first trait we decided to map was a mobility score, and this was in collaboration with Noah's group right here at ASU and Eleanor Carlson at the Broad Institute at MIT and Harvard and UMass Medical School. And the top hit, so this is a Manhattan plot. Each one of these points is a single nucleotide polymorphism. The y-axis shows p-values, and this top hit here was a gene called SH3BP4. And when we saw that, I had to laugh because that turns out to be a negative regulator of the mechanistic target of rapamycin pathway. So this was early. This was just with a couple thousand samples. We know how science works. When we get up to 8,000 or 10,000 samples, this is probably going to disappear. But maybe it'll get more significant. So we're pretty excited about this. This is just the kind of thing that we'll be doing. And keep in mind that we have thousands of phenotypes. So the opportunity to map traits in dogs is just going to be enormous. There's the systems biology. And just really quickly, I'll just show you some pilot data from methylome studies in dogs. So these are actually about 100 dogs from Texas as a pilot that aren't actually dog aging project dogs, just to show that, as in humans, we can predict age. with the methylome, and this was work, 2 graduate students working with Noah and me, Kelly Jin and Elizabeth Goldman at Oregon State did this work. And this is showing actually 2 different measures of the epigenome. This measures methylation sites. This measures chromatin openness or access. Both were used to predict age. So we're going to have metabolome, epigenome, microbiome, flow data. And with all of these, among the many other things we're interested in, it'll be really cool to see if we can use these different domains to predict age in dogs and to predict health in dogs. So I showed you that we built a metabolome clock for flies. The very first set of samples that we collected for dogs aren't large enough yet to give us the power to build a clock, but we do see a signature of age in the metabolome. So that was reassuring. This is the second principal component. The first principal component turns out to be a measure of sample quality, degree of hemolysis in the blood. So final thing I want to say about the dog aging project. So I mentioned the triad randomized control trial. So this is asking whether rapamycin can make dogs live longer and healthier. So our primary goal is to maintain safety above all else. So we have an animal welfare advisory board, which is independent of our project. We have a data and safety monitoring board that was constituted by NIH. So we're running it just like a human clinical trial. It's a double-blind, placebo-controlled study, so we don't know whether the dogs are on placebo or rapa. It's a very low dose. We are erring on the side of too low a dose, again, because we want to ensure safety. 70% of Americans consider their dog to be a member of the family like a child, and so we really want to be very mindful of that. No one is forced, of course, to enroll. The only people who enroll have asked to enroll, and they can quit anytime they want. So all dogs are welcome with the proviso that they have to be sterilized, middle-aged, and large breed. And the reason is if we have 2-year-old Chihuahuas enrolled, we'll never finish the study because they live too long. And then the last thing I want to say about the study. So this paper came out last year, earlier this year, sort of announcing the study to the scientific world. And really our focus of this paper was that this is an open science study. This is a snapshot, a picture of the Terra platform at the Broad Institute at MIT and Harvard. It's kind of a wrapper around the Google Cloud. All of our data are in the Google Cloud, other than the human data, which are not released. Those are all protected. And anyone can apply for access to these data. If you just go on to your favorite search engine and type in Dog Aging Project Data Access, you can figure it out. And if it doesn't show up, reach out to me or Noah. All of you are welcome to access these data. We embargo the data for a year, so trainees, like the trainees in the room here from the Dog Aging Project, can have first crack at it, and then it goes out to the whole world. So finally, just to conclude, I hope in this brief time that we've had together, I've been able to convince you that the metabolome is actually a really powerful lens onto our understanding of how the genotype shapes complex phenotypes. But it doesn't just help us to find the genes. It actually has the potential to offer mechanisms, because the metabolites and lipids that make up the metabolome are, by definition, in many cases, those mechanisms. But I want to stress that we are really at early days. We barely know how many metabolites there are in humans. Most metabolomics experts, biochemists, which I am not, will tell you that there are maybe 10,000 features, but then there are some like Dean Jones at Emory who believe that there are more than 1 million, and it's up to us to discover them. I do believe that there are likely whole pathways that are functioning in us and other pathways functioning in fruit flies that we don't yet know exist because we haven't gone looking for them. As I mentioned earlier, unlike genomics, we will only find what we look for, at least with current metabolome technology. So I think the opportunities for discovery, really fundamental discovery, are massive in this area. So Major caveat, there might be tens of thousands to millions of features. Most are unknown. We haven't even begun really to think about the tissue specificity of those pathways. And it's still currently relatively low throughput, relatively expensive, depending on where you go, between $100 and $500 per sample. So I think the technology still has a ways to go. And then finally, I just want to acknowledge my lab that's done a lot of this work, especially Ben Harrison, and former members, Jessica Hoffman and Mitchell Lee, who did a lot of the work that I presented to you today. The Dog Aging Project is a team study. There are more than 80 people who work on the project in addition to the 41,000 participants. They're pictured here. So the Dog Aging Project is not just Noah and me, but really an amazing group of people. And the other trainees who are on this picture. And we always are welcoming more graduate students, postdocs, collaborators. NIH funds this work, and I thank you for your attention.
All right, so if anyone has questions, raise your hand. I'll give you the mic. And those of you who are joining us on Zoom, if you send something through the meeting chat, I can read it out loud.
It's really great work and touches on so many different things. But I'm wondering about in the Dog Aging Project, if you're collecting, what kind of outcomes you're collecting besides longevity? Are you getting cause of death? Are you getting, for my case, cancer?
Yeah, great question. So we actually have several different ways that we're capturing health data. So the survey, and I will say if you go to data.dogagingproject.org, you'll see the first year survey summary data. So we're now in year 3, but you'll see the kinds of health information we collect, which include owner-reported cancers, including the type of cancer, location, so on. We invite all our participants to upload an electronic medical record every year. Many veterinarians use electronic medical records. Fully 50% of all of those participants have uploaded at least one electronic medical record, and so those are also available for access. And then the third thing that we do that turns out to be pretty interesting is we created an end-of-life survey. So in the veterinary world, often dogs are diagnosed with a terminal disease, and for many different reasons, the owner will elect not to treat, and the dog will be euthanized. Sometimes the owner will bring the dog home, and the dog will be kept alive as long as its quality of life is worth living, and then they'll have a veterinarian or vet tech come to the home and euthanize it. But there's no record of what actually led to the dog's death other than euthanasia. So we created an end-of-life survey, and the interesting thing was that people were so excited to fill this out. These people really want to tell their stories, to share their stories. We even had people not on the Dog Aging Project who asked if they could fill out the survey, and we said, you know, it's not going to be part of our database. And they said, we know, I just want to tell the story of my dog. So so there's the medical information that we get on the MRs. There's also some really interesting sort of sociological information for people interested in qualitative studies, I think, about how we understand the end of our dogs' lives.
Thank you for the, is this on? Thank you for the talk. That was really great. Do you have any plans to share this information with the veterinary groups, or have you had any veterinary groups reach out to you to understand how to better recognize and understand normal age progression in dogs? Because it does feel like they're just kind of like, yeah, the best thing would be to put them down. Otherwise, it's going to be a lot of research to find out.
Yeah. So we have so our chief veterinary officer is Kate Creevy, and really the study started when Nathan Sutter's paper from the Ostrander group came out in 2007. I immediately sent an email to friends at the University of Georgia Vet School, and I got the runaround. And finally, someone said, "Give it to Katie Creevy. She's new." And so she was a new veterinarian there. She reached out to me and she said, in characteristic Kate Creevy fashion, "I don't know anything about aging. I can't help you. I'll meet with you." And she's now the chief veterinary officer, and so she and I really built this study from the ground up. And she's created a group of an advisory veterinarian group of veterinarians around the country. They're very excited about this work. And the work that she's leading, she leads Project One. And from her perspective, that the exact target of Project One is to develop models of normative aging that we can then give over to the veterinary community to be able to do those things. So I'll just give you one specific example. So just like Linda Fried at Columbia and others have developed frailty models for predicting normal aging as a function of different measures of frailty in humans, we're trying to develop a frailty instrument in dogs that a veterinarian could fill out working with the owner. And then the veterinarian could say, here's what we can expect. Here is our predicted lifespan. And here's, really a meaningful metric of quality of life for dogs.
I hope my vet, my future dog, reads the study. That sounds awesome.
Ben, wave the mic somewhere. Oh, wait, sorry, Cyril, go ahead, sorry.
Hi, is this all? Hi. Thank you. That was a cool talk. I just had a question about the metabolomics work in the Drosophila. So I don't usually work with flies, so this may be a silly question. Do we expect the same relationship between genotype, phenotype, and metabolomics if we don't use inbred lines? Because I was wondering how, because if you inbred so much, your effective population size drops.
Yeah.
And so do we expect, like in natural populations, for metabolomics to be the same bridge between genotype and phenotype?
That's a great question. So it's not a silly question at all. We've talked about doing this experiment in flies to ask how, so we can very easily in the fly create a whole range of homozygosity, a whole range of inbreeding from 0 to 1. And we've talked about doing that and looking at how that impacts aging. It's just another one of the studies on the back burner. The cool thing about the dog study is that we have half purebred and a half mixed breed dogs. And among the mixed breed dogs, we have F1 dogs that are the offspring of 2 purebreds. And then we have mutts like Frisbee. And so we will be able to answer that exact question. So I think it's a very exciting question, actually.
Yeah, great talk. So I had a question about IGF-1 and reproduction in dogs. So, natural selection is normally optimizing reproductive success, not longevity. So do you see that dogs that have higher IGF-1 on average have more offspring?
I don't know. That's a good question. What I will say is that with the dog, it's a little problematic. So first of all, the variation that I showed you in that picture of all the different breeds, most of that variation just dates back to the Victorian era. So there are ancient breeds, but most of our breeds are not that old. And in the course of selecting all the different breeds, the sort of metric of success typically has not been number of offspring. It's been, does it look like the dog that I want to see? And that in turn has created some of the problems that we see in dogs. The fact that Cavalier King Charles Spaniels have a ton of heart problems, but they're an awfully cute dog. And the breeders didn't care about the heart problems. They cared about that it looks like a dog with an apple on its head. So it's a really good question, whether independent of that, it's true. And there may be some work that I'm not aware of. There are some people who've been working. Kimberly Greer has done a bunch of stuff measuring levels of IGF-1, and I don't know if people have looked at the relationship between fecundity and IGF-1. So typically, when we downregulate the IGF-1 pathway, we extend lifespan, but at the cost of reproductive success. The other thing that I'll point out is that it's a challenge for our study. 95% of our dogs are sterilized because we reflect, in some ways, dogs in America. We only currently allow 1 dog per household. The people who tend to have intact dogs are often breeders with lots of dogs per household. And if they can only enroll 1, they're not so interested. If they could enroll 5, they would be. So we're talking about opening that up.
You show this very interesting plot of longevity according to different genetic strain of Drosophila. And I was wondering whether on those strain, whether there is like a trade-off between longevity and fertility, for example. And because if we think of the life history theory, we know that longevity is only one trait, and it's usually trade-off with other traits. And so if we think philosophically, like we want to maximize lifespan of dog or humans, but it might come at some other cost for fertility, for example. So I was wondering, yeah, if we observe that in Drosophila, and what's your original input about that?
Yeah, so there's not a lot of information on that. We didn't measure fertility or fecundity in this study. Years ago, I wrote a little paper analyzing data from another paper that claimed there was no trade-off, showing that there does indeed look across species to be a trade-off between lifespan and reproduction. But across species, there's surprisingly little data in Drosophila. Within species, there's quite a lot of support for trade-offs, for fertility and fecundity trade-offs and survival.
Well, thank you, Daniel, for a wonderful talk.
Thank you.